So, good evening to all of you. Today's esteemed speaker, Dr. Shimon, Chairman, Chancellor, and distinguished participants. It is a great pleasure to welcome mention the outstanding a plant physiologist of India, Professor Uday Kumar, Division of Plant Physiology at the University of Agricultural Sciences, Bangalore. And he is actually outstanding a plant physiologist in this country. And unfortunately, we received the sad news that he left the abode of the heaven. So, Hebrew University, Israel. He is currently working as a full professor at the French Associates Institute for Agriculture and Biotechnology of Drylands, Ben-Gurion University, Israel. The main focus of his research is whole plant ecophysiology and accumulation to abiotic stress. His present academic activities are the role of a dew in desert plants, role of root orders in the water and nutrient uptake, carbon and water relations associated with rain and temperature gradients, ecophysiology of allopathic plants and their use in constructed wetlands, reintroduction of and physiology of historically agro-beneficial plants, the role of pesticides in as growth regulators, multidisciplinary study on grape vines including plant physiology, metabolic profiling and spectroscopy. He has guided 38 MSc and PhD students and around 13 researchers worked in this laboratory as visitors and postdoctoral fellows. He published 93 research papers in high impact journals and 4 book chapters and 3 scientific reports and technical papers. He has 2 US patents. He has citations of 3,987 with a H index of 32 and I index of a 68. So Dr. Shimon biodata is too big. So I have his biodata. It's almost running into 26 pages. So it will be very difficult to read the entire biodata. So with this, I stop here. Now I request Dr. Shimon to deliver a lecture.
So thank you very much. I just want to make sure you hear me. Do you hear me? Hello?
Hello?
Do you hear me?
Yeah, we can hear. It's very clear.
Good. So okay. So if there's any problem, let me know. Thank you very much for this kind invitation. Topic we'll talk today about below ground root interaction in crops, and I will relate it to abiotic stress. To start, so we know that the roots are the hidden half, and in order to promote agriculture worldwide and to begin our second green revolution, because we know the first green revolution got to a plateau. So crop yield now is in a plateau and is not increasing. So we believe that studying the hidden half will have a major impact on getting towards this goal of the second green evolution and increasing our yields per area. And this doing in a sustainable manner, of course. So let's talk about roots. So if we look at the below ground, we talk about the rhizosphere or the below ground. So we have lots of root interactions and between either two individuals or and much more is happening in this rhizosphere. If it's bacteria or exudates coming from the roots. And the knowledge that is known about all this is relatively low, not high. We have to always remember what's happening in agriculture. So most agriculture breeding until to date is investing in the upper area, because what we eat is usually, except for a minority of root crops, what we eat is the above ground, either it's the fruit or the yield. And we have to understand that in nature, a natural grass, let's say in this case, will have much more roots than a breeded agriculture grass, which will have much less roots. This breeding for less roots also has an effect on increased yield, but also has major effects on crop tolerance and susceptibility to abiotic and biotic stresses. Because during cultivation and breeding process of both natural and genetic, in my opinion, the breeding, the natural breeding is also a genetic breeding. So I don't see any difference between a regular tomato to a GMO tomato, but this is my opinion. But anyhow, this breeding caused a very large decrease in the root system. And we have to study, if we want to know what's happening between roots, looking at crops and roots.
Shimon,
yes.
The screen is not clearly visible. Can you make it bigger?
The screen is not visible.
Yeah, it is very small.
Screen is visible, but full screen we would like to see.
Yeah, maybe.
In mine, it's full screen. I don't know. I can stop sharing and share again.
Yeah.
What? I was in full screen. So
the bottom there is this slideshow mode. I think that is to click.
Is this better? Is this better?
Yeah. Yes.
No, what's the same?
Yeah. This is very, now it's clear.
Okay.
Thank you.
Okay. So the idea was that we know that agriculture really affects it, and if people are thinking rhizosphere, because this is one of the goals, so people are thinking of many factors, including bacteria, rhizobacteria, looking at the exudates, and looking at the root interactions, either with other roots or with other organisms. And in order to understand how to engineer this, we first have to clearly understand this whole rhizosphere, which is a big enigma in many, although there's lots and lots of progress, there's still lots to learn. So I'll show you some things we do in the lab, which I think are very nice. And this is how it is. So the first topic I'll talk about will be the interaction between roots, root interactions. Not root microbe interactions, but root interactions. And we know that already in 91, Mahal and Callaway showed that if you have two individuals, the root system will grow a little bit differently when there is one individual. So the question is whether this red root knows that this blue root is not himself, it's another individual. And this was very exciting to understand that plants have some knowledge of the existence of the neighbor around them just from the roots. In ambrosia root elongation after interplant, but not after intraplant root contact. And this suggested that this detection mechanisms involves a capability of non-self recognition. So we're talking about a plant, whether it knows whether this root knows that this root is the same self, or whether it's a non-self. So the first study we did looking at the peas, and this like a model plant, and from there we went to other crops. So what we did is we took 2 peas, and we have the roots. So if these are the red here, so if a red sees a red itself, if a pink sees a pink, or a purple sees a purple itself, but when a purple sees a red, it is non-self. So this is the thing you have to understand in the whole setup. Is this clear? I just want to make sure this is clear and then continue.
Yeah, it's clear.
Okay. So this is how the roots look when we have a CAT scan below ground. And when you give this talk, in Austria, you talk about root waltzing. I thought that in India, I could talk about root couples yoga. But in any case, we understand that these are root social interaction. Yes? So the question is whether we have these social interactions and what are the social interactions, how they come to, and there are many, many other questions. How they affect the plant, how they affect the yield, and these are the many questions that we should, this is a thing that I'm talking about. So in this case, we took peas and sativum, and we split the root into 3, we put 3 individual in 3 pots. So in this case, we have 3 individual, in each pot we have an individual, a pea plant, and this is all together itself. In this case, in each pot, we have roots from 2 different individuals. So each pot here is a non-self, okay? Anyhow, in the number of plants, this is 3 plants here and 3 plants here. Now, density-wise, it's the same density. The only difference is that the root of a different individual, they see each other. Okay, so this is self versus non-self. And this is how it looks. We grow them in these small containers. And we measure and we see, let's see, root length is severely affected when you have non-self. NS is non-self, S is self. So under non-self conditions, root length is decreased. Also photosynthesis of the above ground, and the above ground is all the same, is significantly affected when you have neighbors in the roots. So we decided to look at respiration. We know that respiration is a large fraction of all carbohydrates that the high plant assimilates. They are expended in respiration. Up to 70% can be of the carbohydrates from photosynthesis can be expended in respiration. And it produces ATP, energy, reducing equivalents like NADH and NADPH, and carbon skeletons. And we have this very unique system to measure root respiration, and we measure simultaneously both O2 influx and CO2 efflux. Okay, so this is how it looks like. We close the plant, and we seal them, and we move air continuously. And then what we got was really, really exciting, that we saw that in non-self plants, root respiration increased 30% in relation to self. So plants, when they have a neighbor next to them, they're breathing very fast. Like it's very similar, let's say, to the social behavior of humankind. When people close to each other, they usually, if a mouth is close to them, they breathe harder. When they go further, they breathe slower. And this respiration is breath. And the respiration is 30% more when you have non-self. So this was really, really exciting news. And we saw it, you see, this is during a night and day period, either in O2 uptake or in CO2 release. We see the same thing. In black is non-self. Non-self is respiring more significantly, 30% more than self. Okay, so the tentative conclusions for that is that root length area, specific root area of non-self plants decreased significantly as compared to self plants. Root respiration increased by 27% in non-self plants as compared to self plants. And the question is, what happens under stress conditions? Okay, what happens under stress conditions? So this is the root length under control. When we take under salinity 100 millimolar NaCl, we see that non-self plants are not affected by salinity. So this led us to speculate that this being next to a neighbor is reducing your root length and your photosynthesis, but it is at the expense of acclimating to future stress. So the plants have high respiration, which engages lots of ATP, and then this causes them to be acclimated to future salinity stress. And this we could see also in the shoot biomass. And when we look at respiration, so the respiration increases also in the non-self under salinity, but the increase is significantly less than in the self. So plants in under salinity, when you have a neighbor, they invest more in the proportionally more in the biomass than in respiration. So to summarize this, so root respiration in response to competition. So non-self plants were less affected by salt stress than self plants, indicating a possible acclimation to future stress in non-self plants in a feed-forward acclimation mechanism. And these results provide new information on root respiration, which indicates that carbon emission through root respiration may have been underestimated by up to 30%. So the next question, okay, we looked at the same species in peas. What's happening in other crops or in crops? So this is a study that is being done in the last year in my lab, or less than a year in my lab. And the idea is kinship effects of self-non-self interactions. And what is kinship? Kinship is the state or fact of being a kin, a family relationship. And the question is, what kinship did we look at? So we started with looking with one family, the Solanacea. And within this family, we looked at different distances. So we look at three species. We look at cherry tomato, which is an Israeli invention. We looked at field tomato, in this case M82, and we looked at bell pepper. Okay? And the question is, what is the effect of this kinship on the self-non-self interaction? So what do? We do several ways. We started with rhizoslides. So what we do, we grow them on paper like this, and then we could take pictures of the root system on a daily basis and study the architecture dynamics and the growth dynamics of this root system. So we believe that the architecture itself has major impact on the plant tolerance and on the interaction with other plants and bacteria. Now, I will show you some results, but not all the results, yes? But this is what we do. We transfer to the slides, we monitor, and we analyze. And in this case, we had the combinations. We had the cherry, we had field with field, bell pepper with bell pepper, cherry tomato with field tomato, cherry with bell, and field with bell. So inside this, and we did under control and under salinity. Okay, and this, the big advantage of these rhizoslides, so it lets us do very large scale studies in a small relatively area. We could have many plants. You see, it doesn't take lots of space, and it's relatively short term. We do it in short term. The idea is the next step is to go to the greenhouse and then to the field. So we understand that this is not a clear representative of the field studies, but we know that anything we find here is something that is within the genetics of the plants. So it doesn't matter if it's exactly like in the field. Because also if we go to a field, every field behaves differently. depending on its microclimate and environment and whatever. So the idea is if we see anything, we see the genetics of the plant to behave somewhere. So this is the combinations we did, and this is large scale studies. And I'll just show you some results. So if we look at root length, for example, so this is when cherry sees cherry. So under salinity, the root length even is increased when cherry sees cherry. Okay, this is all non-self. It's not self. In all here we have only self, non-self. Under control versus under salinity, and so they behave differently. And if we take it and we summarize this, so we show here, I show here in the y-axis the fold change in root length when, in this case, cherry tomato. When cherry tomato sees a cherry, a field tomato, and when cherry sees a bell, so it's increasing relatively when to cherry. But when cherry sees a bell under salinity, root length decreased significantly. Okay, so this shows us clearly that there is, first of all, kinship recognition. So when cherry sees a field or cherry, it's completely different than when a cherry tomato sees a bell pepper, especially under salinity in the case of cherry tomato. So this is super interesting. Yes, and the next thing we want to know is really, and this is what our goal and aim, how do they recognize, how does this cherry tomato knows this is a cherry, this is a field, and this is a bell? What are the signals? And we know this is not an easy task, but this is what we're trying to do. So just to show you two more examples here. So when a field tomato sees a cherry, it behaves exactly like if it is its field, but when it sees a bell tomato, it is increasing its root length significantly. So when a field tomato is next to a bell, the root length is increased significantly. And this is actually was our hypothesis that plants will behave like that. When it sees someone who is further related, not closely related, it won't have that much effect of non-self because they compete for different resources. So in this field tomato, this is exactly in the root length, this is exactly what we saw. In the bell pepper, we see that again, under control, it's good for them to grow with field, but under salinity, it's always bad for them. So here, this is like to summarize these results. So in the control, when field or bell pepper, they both enhance their root length, okay? Cherry, when it's with field or bell pepper, is also enhancing its root length, but when either bell pepper or field or with cherry, they are decrease their root length, okay? Blue is positive interaction and red is negative interaction. Under salinity, we see a different by doing grafting of different combinations. And then we'll have different interactions of above and below ground. Yeah. So I think it's a complex system. It's not only below ground interactions, it's below ground and above ground. Yeah.
Dr. Shimon, I have a small, I don't know whether it is a immediate requirement or not, but we have also have a pulse crops where the nodulation bacteria are very common. If a combination of a pulse crop along with a non-pulse crop like Solanaceae, if they are grown together, do you expect any drastic change in its behavior?
Yeah, so the question, I don't know. I think the first thing is to go to the farmers themselves and see what's happening there, you know. But what I expect is when you'll be, this was my hypothesis that was not very supported from our preliminary results, that when you are closer in your kinship, the effects will be higher. When you are further away, so these effects will be less because of the need for different resources, like legumes, let's say, for example. But I really, so our first results here show that, let's say, bell pepper versus cherry, although they're in the same family, they sometimes behave better than when cherry is with cherry. So I don't know.
Because in tropical conditions, usually we recommend pulse crops as an intercrop to enrich soils for the growth of the companion crops, which are non-leguminous. I don't know whether there will be any antagonistic effect or may not be antagonistic effect, but any root companionship or root relationship may vary or not. I think we don't have right now any studies on that, but we know, but for the sure, the nodulation pulse crops will definitely help in enriching the soil properties and in turn the yield also.
Yeah. So this is very interesting for me to see, and maybe to do like, this is another thing to do, to analyze the results that there are out there. The only thing is get these numbers that you're talking about.
Thank you.
I'm not sure.
Any other questions? Dr. Shimon, I wanted a small clarification. See, in India, the grape cultivation is on a dogridge rootstock. But so this dogridge rootstock is highly tolerant to the abiotic stresses like water as well as salinity. Do you think that any other microbes are specifically associated with a dogridge rootstock to impart a tolerance?
Yes, so first of all, I'm sure there are microbes that are related. In Israel, we use a different for grape. We use rogery. Yeah, but for now I didn't do any study about that. So there's so much to study, and we're not there. Yeah, so the idea is they are out there commercially now. People are selling microbes, okay, to add even through the fertigation system. And I believe that there are some. The question is which are exactly. This is a long-term agronomic study.
Thank you. Any other questions? If no other questions are there, I think Dr. Shimon, also an eminent plant physiologist, I think Professor Uday Kumar, before closing the session, I would like to read his biodata for the sake of audience. See, Professor Uday Kumar, born Andhra Pradesh, India. Professor Uday Kumar studied for school education, BSc from Andhra University, MSc from Moscow University, and PhD also from Academy of Sciences ESSR. He joined the University of Agricultural Sciences Bangalore during 1973 and worked there as a professor and head of the Department of Crop Physiology and also INSA senior scientist from 1973 to 2021. He was also visiting professor at Agricultural University, Netherlands. He made a significant contributions in research related to photosynthesis, abiotic stress tolerance, transgenic crop improvement, phenomics, and genomics of important crops. An inspiring teacher, Professor Uday Kumar, guided and mentored a scores of the students. As a chairman and member of a review committee on genetic manipulation, DBT, and genetic engineering appraisal committee, Ministry of Environment, he was instrumental in formulating the policies of a genetically modified organism regulation in India. An unassuming, endearing, and humble personality, Professor Uday Kumar infused a clarity and wisdom in shaping research programs in ICAR, CSIR, DST, DBT, UGC, and several universities. And he was fellow of all the 4 major science academies of India. Professor Uday Kumar passed away on June 25th, 2021. I think so. It is a great loss to not only Indian science, it is also a global science also. So we at the BEST University, because many children lost their parents due to COVID. So we at BEST University are making an announcement that all the students who lost their parents due to COVID, the university provided all the financial assistance. So they did not pay any fees or hostel, everything. We are announcing that because a small gesture of the university, we decided to announce for students, they can get the free education in the university. So we are announcing at this moment when Professor Uday Kumar passed away. I think both are coinciding. So we are really grateful to our management. So who has given a gesture for the students. And if no further questions are there, so I would like to thank Shimon Rachmilevitch. So he has given an outstanding and inspiring, extremely informative lecture, because this is the need of the hour, particularly the entire world is actually struggling for the water, and the entire agriculture will be depending upon the scarcity of the water. The topic is highly appropriate. We are highly grateful to Dr. Shimon for giving an inspiring lecture.